cell division phase is M phase, means mitotic phase. So in this mitotic, number of chromosomes, if the parents is having 23 pairs of chromosomes, then offsprings also same number of chromosomes are present by the mitotic cell division. So these are the main reason behind that. So this mitotic phase can start with karyokinesis and with cytokinesis. So in this mitotic karyokinesis, again they are divided four subphases: prophase, metaphase, anaphase and telophase. And karyokinesis ends with cytokinesis. Now first of all, what is the karyokinesis and cytokinesis here? So division of nucleus, it is called as karyokinesis, and division of cytoplasm, it is called as cytokinesis. Now M phase, it is the actual cell division phase. So what I said, cell is divided and result the daughter cells. So this is the cell cycle. Cell can divided and result the daughter cells. That is the cell cycle. So what is the mitosis? Mitosis, it is the equational division. So equational division means if you see the parental chromosomes, if the number of chromosomes are present in parent and offsprings also same number of chromosomes are present, that is the mitosis by the help of mitotic cell division. That's what this is the equational division. So mitosis, who discovered mitosis in plants? Strasburger. And mitotic cell division, who discovered in animals mitotic cell division? Means Walter Fleming. Walter Strasburger. In plants. What about animals? Walter Fleming. Walter Fleming. Walter Fleming. Animal cell. Animal cell. So in plants, who discovered mitotic cell division? Strasburger. And in animals, who discovered mitotic cell division? By Walter Fleming. This mitotic cell division also we called as homeotypic cell division. Why this mitotic cell division is called as homeotypic cell division? If you see, daughter cells means daughter cells is identical to each other, and mother cells means daughter cells. So daughter cells are identical to each other and mother cells. That's what this is called as homeotypic cell. Homeotypic cell division. Why mitosis division we called as homeotypic cell division? Daughter cells are identical to each other and mother cells. That's what this mitotic cell division we called as homeotypic cell division. Homeotypic cell division. This mitotic cell division we are seeing in plant cells. In plant cells, both haploid cells and diploid cells are present. Now what about animal cell? Animal cells is having only haploid cells, diploid cells only having this mitotic cell division. The animal cells, some animal cell like social insects we can see in haploid cells also this mitotic cell division. So plants both haploid and diploid cells. And animals only diploid cells. Some social insects we can see in haploid cells also mitotic cell division. Haploid cells. What is the experimental material for identification of mitotic cell division? So experimental material for identification of cell division, mitotic cell division, that is the onion root tip. So this is the onion root tip is the best experimental material. Onion root tip is the best experimental material for identification of stages. Cell division start with the karyokinesis, ends with the cytokinesis. So, what is the karyokinesis? Karyokinesis means division of nucleus, we call it as karyokinesis. What about cytokinesis? Division of cytoplasm, we call it as cytokinesis. This karyokinesis, again they are divided 4 subphases. What are those 4 subphases? One is prophase, metaphase, and anaphase and telophase. Prophase, metaphase, anaphase, fourth one telophase. Now we can start one by one: prophase, metaphase and anaphase, telophase. Let us start first stage, that is the prophase. So prophase, our understanding convenience purpose, this prophase again they are classified 3 subphases: early prophase, mid prophase and late prophase. So what happens this early prophase stage? Chromatin material will condense. So chromatin material will condense and form a rod-like structure. It is called as chromosome. This is the chromosome. So chromatin material will condense to form a rod-like structure. It is called as chromosome. So this is the early prophase stage. How the chromatin material will condense? Due to dehydration, chromatin material will condense. Now what happened is mid prophase stage. So after completion of chromosome can split longitudinally except centromere position. So chromosome can split longitudinally except at centromere position to form a chromatids. So late prophase, nuclear membrane and nucleolus and organelles, Golgi or endoplasmic reticulum disappear, means nuclear membrane and nucleolus disappear. And organelles like endoplasmic reticulum and Golgi complex also disappear. So due to disappearance of nuclear membrane, chromosomes can migrate from nucleoplasm to cytoplasm. These are the prophase stage. So what happens this prophase stage? Nuclear membrane and nucleolus disappear and organelles ER and Golgi complex also disappear. Disappearance of ER and Golgi complex. Now this is the first one, this is the metaphase. Next, second one, metaphase. You see that, look on this picture. Complete chromosomal condensation occurs in metaphase. So that's what chromosomes can migrate at the middle. It is chromosomes can migrate at middle. That's what it is showing. It is the best identification for morphology of chromosomes. Means metaphase plate we called as morphology of chromosome. Why we called as morphology of chromosomes? Here clearly visible chromosomes are arranged. So in this metaphase we called as equatorial plate or metaphase plate. Why it is called as equatorial plate or metaphase plate? Chromosomes can move equatorial plate, means chromosomes can migrate migrated to the middle plate. So plate we called as equatorial plate or metaphase plate. Metaphase plate. Equatorial plate. Equatorial plate or metaphase plate. Metaphase plate. That's why it is the best stage for identification of morphology of chromosome. Morphology of chromosome. Now here, each chromosome is having two chromatids, one centromere and two DNA molecules. So each chromosome is having two chromatids, two DNA molecules, and one centromere and two kinetochores. If you see this, two chromatids, one DNA molecules, two kinetochores. Now what is the kinetochore here? What is the kinetochore? Either side small disc-like structure present on centromere. It is called as kinetochores. So either side small disc-like structure is present on centromere. It is called as kinetochore. What is the function of this kinetochore? It is a site. This is the site. It is a attaching of spindle fiber. So this is the site. It is attaching of spindle fiber. So it is the site for attachment of spindle fiber. So that is the kinetochore. Next stage, that is anaphase stage. So what happens this anaphase stage? To splitting of centromeres. To start separation of chromatids. So splitting of centromere we are seeing in this anaphase stage. Then after splitting of centromere can start separation of these chromatids. So separation of these chromatids to form a daughter chromosome. How the daughter chromosomes are formed in anaphase stage? Splitting of centromere can start separation of chromatids to form a daughter chromosome. Division of centromere. Separation of chromatids. Now, daughter chromosome. Each daughter chromosome is having having one chromatid, one DNA molecule. So one chromatid and one DNA molecule. Now here another most important point. What is the location of this centromere and chromatids? Look on this picture. If you see, the centromere is located towards the poles and chromatids is located behind the centromere. This is the location of this centromere and chromatids. So centromere here located towards the pole and chromatids are located behind the centromere. Fourth stage, that is called as telophase stage. If you see the telophase stage, what happened? Telophase is full opposite to prophase stage. What we observe in prophase stage, nuclear membrane and nucleolus is disappear and organelles also disappear. But here reappearance of nuclear membrane and nucleolus and Golgi complex and AR also reappear. And here chromatin. So chromatin material will decondensed due to hydration process. So chromatin material will decondensed into hydration process. That is the nuclear membrane and nucleolus disappear and ER Golgi complex disappear. So that's what cell is having two daughter nuclei at the end of telophase. Each cell at the end of telophase, each cell divided to daughter nuclei. These are the changes of telophase. Next, kinesis. If you see cytokinesis, if the cell division can stop, what happens? We are getting multinucleate condition. So when this means division of cytoplasm, we call it as cytokinesis. So this cytokinesis occurs in nucleus. Centriole duplication occurs in cytoplasm. If you see cytokinesis occurs in plant cell, that is the cell plate. Cytokinesis occurs in animal cell by furrow in plasma membrane. So cytokinesis occurs in plant cell by cell plate and animal cell by the help of furrow in plasma membrane. First one, plant cell plate, animal cell furrow in plasma membrane. If you see the animal cell, it is cytokinesis occurs by furrow in plasma membrane. Furrow which is formed in cell membrane which is grow centripetally can join with the middle cell plate, to divide two daughter cells, So animal cells, cytokinesis occurs by furrow in plasma membrane. How it is? We see the furrow which is growing cell membrane which is growing centripetally and join the center to form to divide the two daughter cells. So these two daughter cells are having equal number of chromosomes. These are the two daughter cells. Now, if you see the cytokinesis occurs by in plant cell through the cell plate. So how it is possible? See, remains of spindle fibers can form a barrel-shaped structure at the center of the cell. So remains of spindle fibers can form a barrel-shaped structure at the center of the cell. And Golgi complex, remains of Golgi complex can releases calcium and magnesium in the barrel-shaped structure. So in this barrel-shaped structure can turns the liquid plate. In this liquid plate can join the parental cell plate. This liquid plate can join the parental cell wall. The cell plate can turn the middle lamella. So this cell plate can turn the middle lamella. So this cell plate can touch the middle lamella. This also we called as phagmoplast. This cell plate or phagmoplast can divided if we grow new individual organisms. can grow the new individual organism. This phagmoplast organisms. So new individual organ. This is the cytokinesis is completed by the cell plate.